Inheritance of two genes. By now you all already know that Gregor Mendel, an Augustinian monk, grew and investigated garden peas and subsequently explained heritable factors. He also posed and confirmed several salient hypotheses and instituted a new vocabulary to explain his findings that is still in use today. There is more than one form for a gene, allele. For example, based on his experimentation with purple and white flowering pea plants, he deduced, after examining the results of numerous crossings, that one gene is responsible for the purple color and a different gene is responsible for the white color of the flowers. In most cases, two genes are needed to express a trait, one from each parent. Likewise, if an organism is true breeding, both genes that control the same trait had to be the same gene, homozygous. The principle of segregation states that during egg and sperm formation, the pair of genes separates, segregates, and only one gene becomes incorporated into either the sperm or the egg, and the other gene can also become incorporated into either a sperm or egg, so that when sperm and egg unite, the offspring has the normal number or pair of genes. When two different alleles for the same trait are found in an organism, the gene that is expressed is the dominant allele and the gene not expressed is the recessive allele. If red is dominant over white in flowers, the offspring between a homozygous red and white flower should all be red. The principle of independent assortment states that each gene from the pair of alleles separates independently during gamete formation. Mendel noticed that during a dihybrid cross of two traits, the offspring inherited the alleles as if they separated from each other and had equal chance of inheritance as the alleles from the other trait. Specifically, Mendel crossed a plant that was homozygous dominant for round, yellow seeds, capital RR, capital YY. Capital R is equal to round, capital Y is equal to yellow, with a homozygous plant with wrinkled green seeds. Small rr, small yy. Small r is equal to wrinkled, small y is equal to green. If the genes stuck together and did not act independently, he hypothesized that the F2 generation must contain the same genotype as the parents, either capital R, capital Y or small r, small y. For reference purposes, the F1 generation is the product of the mating of the original parents. The F2 is the product of the F2 mating. The F2 then becomes the grandchildren of the original mating. Through experimentation, he found the following genotypes: capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. He correctly concluded that the individual alleles were segregating from the paired alleles and were free to unite in all possible combinations. Refer to the illustration F1 Punnett square. It can be used to predict the results of an capital R, capital Y and small r, small y. First, the genotype of the parents must be known. In this case, capital RRYY and small rryy. Next, they are segregated. If they independently sort, the possible genotypes are capital RRYY and small rryy. All of the F1 first generation offspring are heterozygous, meaning they contained both a dominant and recessive gene, capital R, small r, capital Y, small y, round and yellow. However, in the F2 generation, interesting data revealed a variety of offspring. Refer to the illustrations F2 Punnett square. Parents' genotype capital R, small r, capital Y, small y into capital R, small r, capital Y, small y. Offspring phenotype. Round yellow is equal to 9, round green is equal to 3. Wrinkled yellow is equal to 3, wrinkled green is equal to 1. The gametes had to sort independent of each other as depicted. The phenotypic ratio of 9:3:3:1 is typical of a dihybrid cross. Mendel could tell by the phenotypes of the offspring that the gametes had recombined in such a way that they had to have an equal chance of combining with the other allele.